Okay, so today we are going to learn about how sex cells are produced. And by sex cells, we mean the cells that are involved in sexual reproduction. So, lesson 2.2, sex cells and meiosis. Meiosis. I've also sometimes heard it pronounced meiosis, but I think that's ridiculous. Having said that, if that's how you prefer to pronounce it, go nuts. But I was always taught meiosis. I don't know. This is what I'm saying. One of them has to be silent. Now let me remember this. When two vowels go walking, the first one does the talking, the second one does the talking. This was a rhyme I learned as a kid. Unfortunately, I can't remember if it was first or second. Two vowels go walking. Two vowels go walking. I can't remember. You know what the problem is? At some point, my friend was trying to teach me how to read words in German, and they do it the opposite, and now I can't remember which is which. All of which is to say, English is stupid, because what if we just had vowels all made just one sound, and then that was just the rule? Wouldn't that be nice? In which case, this would be like meiosis. Like in Italian, that's how it is. You know what sound this makes in Italian? No. A. You know what sound this makes? E. You know what sound this makes? Say, ah. And this? Oh. Actually, that makes it look like it's oo. Oh. Because this one makes the sound ew. And that's it. That's the only sounds you can make in Italian. Ah, a, e, o, oo. That's it. You know what they make Y say? Nothing. They don't have one. And you know how we have like these both make a s sound? Yeah, they don't do that. That makes a s sound. This makes a ch sound. If it's with an I or an E, and if with an O or a U, it makes a k sound. That's it. And this, no J. They don't have a J, right? They just use G. Anyway, that's your Italian lesson for the day. English is impossible. Unpronounceable. Can you imagine having? Did anyone here not? Is anyone here like English isn't the first language? Is English wasn't your first language? How old were you when you learned it?
I was 8.
So you're pretty old, and like you speak English wonderfully. English is crazy. Like how do you like? I can't imagine. I found it so hard to learn like French and Italian. I'm bad at both of them, but they're both like French is kind of tricky, and Italian's easy. English is crazy. Nothing makes sense. There's no rules. There's no rule. Like we say there's rules, but they're all lies. Can't imagine, yeah. 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 That's so cool. Isn't that amazing? But yeah, I cannot imagine. I can't imagine going to any country and having to learn the language, but then just thinking like we take for granted, but English is like the most messed up, confusing language in the world. It's so crazy. Oh, it's so dumb. Sorry?
I want Thorn back.
Indeed. All right, anyway, let's get to it. So, the basic idea of this is that the cells of organisms that reproduce sexually are diploid. So they have 2 copies of each chromosome. Oh, less common spelling of each without an A, because why wouldn't there be an A in the word each? Thank you, English. And why wouldn't there be an H in the word chromosome? It makes perfect sense. Everything just makes sense. Chromosome. And we say that as being, we say they're diploid. And if we imagine our happy little cell here, there's its nucleus. And if it has 2 chromosomes, maybe these little guys here, then it's got 2 copies of both. Right? Does that make sense? And these ones are chromosomes from mom, so we would say they're what chromosomes? Maternal, very good. And these ones would be paternal. By the way, they're not actually purple and red. I'm just doing that to show. And I'm going to erase nucleus because honestly, you know that's the nucleus. Okay. So then the question becomes, when organisms reproduce sexually, they produce gametes, and these are sex cells with only 1 copy of each chromosome. Examples would be seeds and pollen. Right? Seeds have 1 copy of the chromosome. Pollen has another. When they come together, they can make a plant. When sperm and egg come together, they make a new animal. The important thing is, in both cases, this side here is diploid. It has 2 copies, and we sometimes see that written as 2N. N just for number of chromosomes, so humans have 23 unique chromosomes, so our normal cells have 2 times that many chromosomes, 46. 2 copies of 23. What term do we use for just having 1 copy? You'd think like diploid and like singloid or something, but haploid is the term, that's right. Diploid and haploid, which means just 1 copy. All of the regular diploid cells, which are called somatic cells, are produced by a process you already know. How do you make new cells? How do cells reproduce? By mitosis. But sex cells are produced by a process called meiosis. And then I'll give you a moment to write this down. Don't worry. Why is that? She would, that's true. But again, I'm going to give you a moment. It's good for her that I rush her. She'll thank me one day. That's not true. She won't. But I say that to make myself feel better. What's up? How are we doing? I know. I heard. I love it. Sorry? Yeah, absolutely. Nope, somebody has taken my masking tape.
Do you know who would have in this school?
I bet you Alfred does.
Okay.
I have scotch tape if you need it. What do you bring masking tape on?
We're putting on chargers to label them to the Chromebook.
No, don't do that. No, absolutely.
But,
No masking tape. No. Masking tape's banned on things. You don't put masking tapes on things. You can tell Reeser I object. Print labels. Amelia can show you how.
Okay.
And don't use the paper ones either. Use the shiny. No masking tape. Masking tape is banned. We don't put masking tape on things. God. Let me tell you something right now. If you ever work in a restaurant or a chemistry lab and you're the guy who puts masking tape on things to label things, everyone hates you, just so you know. If you're wondering because it's like, oh, it goes on easy, and then a day later it comes off easy. Yeah, what about a week later? You've ruined whatever you've put it on. I have glassware back there that like this masking tape residue, and I've spent days like trying to scrub it off. It just has like the power of God holding it on there, and I don't know why. So just listen. Don't use masking tape to label things. Oh, it's the ultimate pet peeve. There's somebody who still does it with glassware here, and I don't know who it is, and I'm always trying to catch them and then like politely be like, hey, don't do that. It's a terrible idea, but not be impolite, right? Because you're not being polite, right? So it's awkward. But yeah, no, don't do it. No masking tape. Anyway. So I'm going to give a really brief version of meiosis, then like a moderately brief version of meiosis, then like a full-fledged version. Does that make sense? We're going to kind of do it at 3 levels of complication. So have you ever seen that kind of as a model of teaching things? There's actually like a whole series of like YouTube videos of this, which are interesting, where they say like, you know, expert explains this at like 7 levels of complexity. But it's kind of a good way to approach things sometimes. So are we ready for our first level of complexity? At the most basics, that's weird. At its most basic, we'll look at like this. So meiosis, difficulty level, easy. All right. So let's talk about meiosis viewed at a difficulty level of easy. And it basically goes like this. We have a normal somatic cell. It has a nucleus, because what kind of cell is this if it has a nucleus? It's a eukaryotic cell. Good. And we have 2 copies of each chromosome, right? We've got a little chromosome there, and we've got the big chromosome and the little chromosome there. So we start with a normal cell, all right, which is to say it's diploid, which is to say it has 2 copies of each chromosome. And we undergo what we call meiosis 1. We go through meiosis a first time. Right? And when we go through meiosis for a first time, what happens is that we end up with 2 daughter cells, but not in mitosis. What would this look like? The 2 daughter cells would look exactly like the identical one. And if you remember, at the beginning, it's worth mentioning that this here, these chromosomes are full of duplicated DNA. Chromosomes are duplicated. And what's it mean to say they're duplicated? That's why they're shaped like an X. It means that they're stuck to a perfect copy of themselves. Does that make sense? So you actually have 4 copies of each gene in here right now, because this chromosome right here is X-shaped because it's basically been photocopied, right? And remember, that's normally what would split in half. Do you remember that in mitosis? Normally that X splits in half. Well, what happens here is that it doesn't split in half. In fact, we get 1 copy of the big one and 1 copy of the little one here, and 1 copy of the little one and 1 copy of the big one here, still duplicated. So instead of pulling the chromosomes apart, it actually pulls half of them to one side and half of them to the other, still duplicated. So still duplicated, but only 1 copy of each chromosome. In other words, we say now already these two cells are haploid. Does that make sense? Now, this could be a sex cell. That could go off and be a sperm. The problem is, yeah, it's only got one chromosome 1, but chromosome 1 is currently stuck to a photocopy of itself, which is almost the same thing as having two copies of it, isn't it? Does that make sense? You couldn't do this and make new organisms because every time each chromosome would get twice as big, and that's not sustainable. Does that make sense? So now we have to undergo meiosis 2 and actually split those in half. Which means how many cells am I going to end up with? This one's going to split in half, and that one's going to split in half. So we're going to end up with we have two cells, and they're both going to split in half. 4. Very good. So we end up with 1, 2, 3, 4 cells. Each of them has a nucleus. And each of them has, so this one still has one copy of the big chromosome and one copy of the little chromosome. And this one has one copy of the big and one copy of the little, but now they've been torn apart from the duplicated cells. Does that make sense? Yep. So this is not duplicated, and it's only one of each chromosome. In other words, it is also haploid. And these are then what we would call proto or pre-sex cells. They actually will undergo some modification, and a few more things will happen to them before they become sex cells. For example, if they're going to become a sperm, then they're going to have to, like have tails and things like that. And if they're going to become eggs, they have all sorts of things like that. So these are then ready to be processed further into sex cells. I want to make sure you have a minute to write that down, and I want to talk our way through it and just see how it's different than mitosis, which don't write this, okay? Unless, I guess if you really want to. But if we compare it to mitosis, in mitosis, we start with one cell that has two copies of each chromosome. And all that happens in mitosis is that splits in half, but the chromosomes split in half, and so we end up with still two copies of each chromosome, just not duplicated anymore. Does that make sense? That's what happens in mitosis. But in meiosis, we get this strange first step where first they get taken still duplicated into the opposite ends of the pole. We make a new cell. And then from there, in the next step, they get split apart. Does that kind of make sense? This side? The other side. Yes. How's that? I'm sorry. I will write it more neatly. No, you're not blind. I'm messy. And you can say it. It's okay. Still duplicated. Sorry, I do the thing where I start cursive writing, and I don't mean to. Right. Yeah, take a minute. I don't want to rush you at all. Why does everyone keep saying this to me? Who has fired me? I didn't quit, I'll tell you that much. No, I was planning on coaching. Oh, Did they say that? Have I been ousted and I'm not aware of it? Nobody's told me I'm out? Listen, I'm hoping to, but if somebody comes up to me and tells me, like, hey, you're out, then I'm out. I don't know. I do what I'm told. Unless I don't feel like it. But I'd like to. My plan is to coach. I'm hoping. I really enjoy coaching.
Are we going to start soon?
I would like to. Yeah. There's people going around saying that we should be able to start practicing like next Friday. I told the girls, like, I'm happy to run practices ASAP. Like, obviously, like,
Practices or tryouts?
well, like practices for now, and then we'll have to do tryouts once the season officially starts. But if people just want to get in and just start warming up and having fun and playing, I'd like to do that as soon as possible. So yeah, somebody had mentioned as early as next Friday. I'm all about it. I think that'd be great because it'd be nice to have some reps in and like be playing before the season actually starts. And like even if some of the boys from the guys team want to come out and like practice as well on an extra day a week, like that's great. I don't think we want to run like a full boys practice and then just a few girls, but I think like we used to do last year where you get 3 or 4 guys out. I think that's a good fit. So no, I'm excited. Everyone keeps saying this. Is the new guy like Orlando's out? I'm in because like that's cool. I can live with that. He's lovely. His name's Mr. Ellis. Good guy. Or at least I thought he was a good guy. Maybe he's stealing my job and not telling me. In which case, fie on you, Mr. New Guy. But I don't think so. I think it's a miscommunication. Because yeah, my hope is to coach. I like coaching. Okay. Now, I want to look at this again. But here we just kind of hand waved and said this happens, and then we hand waved again and said that happens. But we're going to look through the stages again. Because really, meiosis is just mitosis happening twice back to back. And there's one little change. There's one little change that changes kind of what happens. And after that, it's just mitosis. So let's try it again. Let's try meiosis again. This time, difficulty level medium. That's not at all spelled right. Meiosis, difficulty, medium. I shouldn't even say difficulty because it's not difficulty. It's more just complexity. How are we looking at it? Recall the phases of mitosis. What are they again? Prophase, metaphase, anaphase, and telophase, and then cytokinesis, right? Cool. So let's take a look at that. We start with interphase, right? And interphase is just the cell doing its happy cell thing. There's the cell. There's its, and we're just going to stick with this idea that it's got 4 chromosomes, 2 of each. I've been using purple. I guess I'll stick with purple. And in interphase, if you remember, the last thing that happens is the cell takes its DNA and it makes a perfect copy of it, right? And now, normally we would enter mitosis, but instead we're going to enter meiosis 1. Meiosis part 1. And the 1 just means that this is going to happen twice. Okay, and we're just going to draw this out. What's the first step in mitosis or meiosis? Prophase. So this is going to be prophase 1, because it's happening for the first time. And in prophase 1, we're going to have the nucleus start to dissolve. And what's going to form at the poles of the cell? Yeah, which come out of the centrioles, right? So it's going to start to look like that. We're going to get the nucleus dissolving. We're going to get the centrioles and the spindle fibers. I'm not going to label everything here. I will eventually give you a handout that has things labeled. I don't want this to be messy. I want to just kind of be able to see it. Does that make sense? What comes after prophase? Metaphase. In metaphase, the nucleus is totally gone. Where are the chromosomes now by the time we get to metaphase? right in the middle of the cell, right at the equator, we call it. So we'll have the cell, and now we have big X, little x, so that's chromosome 1, chromosome 2, chromosome 1, and chromosome 2. Remember, the reason we have two chromosomes 1 is that one of these came from this cell's mom, and the other one came from this cell's dad, right? And chromosome 2, one came from its biological mother and one from its biological father. And by now, what is true about the spindle fibers? They've connected, yeah. But there's this thing that kind of happens, which is, and actually I shouldn't assume, in fact, I'm going to have it so it matches the earlier portion of the note. We're just going to kind of go this way and say, actually, these ones get taken like that. They connect up. And now, what comes after prophase, then metaphase? Anaphase. And this is where the difference happens. Because in mitosis, what would happen to these chromosomes now? They would all get split apart. So the duplicate would separate. But here is where the difference happens. Which is that here, this centriole pulls, and it pulls a couple of the chromosomes in, half of them specifically, and this one pulls half of them in, and they're still duplicated. And here we get the big chromosome there and the little one there, and we'll talk about which one it takes and which one it doesn't at a later time. But note, this is our first difference. The chromosomes don't split. Instead, one copy of each goes to each pole, still duplicated. Take a pause at that. Take your time and write it. Yes, absolutely. And do I need to slow down a bit? I need to slow down a bit. I'm going to go make a cup of tea so you've got lots of time. Is that zoomed in enough so you can see that? Yeah? Okay. Sure. would help directly if we use the distiller. It's kind of better to have two people on it. I think I would like to get like a little picture. I mean, do what you're told. But... All right, how are we doing? Now, obviously, we have prophase, we have metaphase, we have anaphase. In prophase, it's just like mitosis is normal. Nucleus dissolves, centrioles produce spindle fibers. Metaphase is pretty normal. They line up at the poles. But then in anaphase, what happens is that instead of splitting, whole chromosomes are pulled to each side. And so we end up with half as many on each side as normal. Now, still stuck to the duplicate, right? Still X-shaped, but now half as many. Now, what comes after prophase, metaphase, anaphase, and then telophase, right? And in telophase, we get the formation of a new nucleus and the cleavage furrow, right? And we get that formation of two new cells. And when we do that here, on this side, we're going to have one copy of each chromosome, but stuck to its duplicate. And on the other side, the same. And then the outcome is two daughter cells. And the daughter cells each have one copy of each chromosome. What do we call cells that have one copy of each chromosome again? We say that they are haploid cells. Good. And sorry, I wrote telophase, but of course I should have written telophase 1. Because it's all going to happen a second time. All right, take a minute, make sure we've got that down. Why can't we stop here? Why can't that just be a sperm cell or an egg cell? It only has one copy of each chromosome. In this case, chromosome 1 comes from mom and chromosome 2 comes from dad. In this case, chromosome 1 came from dad and chromosome 2 came from mom. It doesn't have to be that way, by the way. It can kind of happen randomly. But what's the problem with these chromosomes? They are still... Yeah, and we say whole, really they're stuck to a copy of themselves. They're duplicated. Does that make sense? So these cells are haploid, but each chromosome is still duplicated. Which means we enter meiosis 2. Now, guys, we have a collective choice. This is going to happen to both of these cells. Do we want to draw 2 cells at every stage here? Or do we just want to draw it once and know that it happens to both of them? What do we think? Kind of think drawing both of them as actually helpful, annoying as it is. In fact, that's what I'm going to do, though. If you want to draw it just once, go nuts. The good news is, by the way, when we're done this diagram, we're done for today. So now we enter prophase 2. Where are 2 daughter cells that we made above? What's going to happen in both of them? The nuclei will start to dissolve. What will form at their poles? Centrioles, and they will start to produce. Centromere is the middle of the thing. Spindle fibers, yeah, good. And I haven't drawn it yet, but of course, inside the dissolving nucleus, there's going to be the chromosomes. Okay, what's going to come after prophase 2? Metaphase 2. I'll give you a minute to draw this. I'm not sure. I'm going to see if. Of course, it won't be. It's not going to be ready yet. Don't boil 4 liters of water in 2 minutes. We got that down? Okay, after prophase 2, what's going to happen? Metaphase 2. And by the way, this one, there really aren't any differences. This really does just happen the way mitosis normally happens. So in metaphase 2, what's it going to look like? I'm going to draw both these. There's daughter cell number 1. Oh, okay, I hate that. There's daughter cell number 1. There's daughter cell number 2. And the nucleus is totally gone. Where will they be? Right in the middle. And there we are. And what else isn't going to be true? What do they look like in metaphase? They're lined up at the middle. Where are the spindle fibers now? Yeah, they've attached at the centromeres. Good, very good. There we are. I hate drawing spindle fibers. I think mine always looks stupid. There we are. Okay. And then, just like in mitosis, after prophase, metaphase, we get anaphase. And this time, anaphase behaves the way it normally does, which is to say, what's going to happen? it splits it apart. Okay, and then after anaphase 2, what will we get? Going too fast again, aren't I? So I just get excited. I find this stuff exciting. Everyone's like, yes, me too, Orlando. I also find this very exciting. I think you do. Even if you don't quite realize it yet, I think you do. Because it's fascinating. It's happening like in your cells right now. So after anaphase comes telophase, telophase, whatever you want to say it. And that's where each of these is going to begin to kind of pinch in and separate into itself. And what are we going to get forming around each pair of chromosomes? A new nucleus. Bless you. Of course, I wrote telophase, but what should I have written? Telophase 2, or telophase, if that's how you pronounce it. I'm never quite sure how that one's supposed to be pronounced. And then finally, what do we get? And at the end of cytokinesis, what do we have? 4 cells, and each of them has a nucleus, and each of its nuclei contains only one copy of each chromosome, not duplicated. In other words, what we finally have is a haploid set of cells. And there's 4 of them. That's it for today. I feel like that's a lot. Does that feel like a lot? Feels like a lot to me. We'll take some time and sit on that. But that's how sex cells get made. And of course, this happens. This happens in the urogenital tract of males and females. When does sperm production happen? In, like, if we're talking humans, sorry? Yeah, it begins during puberty and carries on until death. Yeah. So, yeah, basically, males produce. Usually there's like a dwindling in efficacy, but yeah, like, sperm counts sort of drop as men age. It sort of peaks in the like teens and 20s. But what about females? When are eggs produced? Yeah, in fact, it's really weird to think, but like, this is really weird to think. But the egg that became you was actually like already produced in your mother before she was born, which is really weird to think, right? Which means that the egg that became you was actually at one point like inside your grandmother's abdomen, right? Inside grandma, inside the fetus that would become your mom was already the egg that would become you. A woman is born with all the eggs she'll ever produce. Women don't produce new eggs through their life. They just release them, all right? With each menstrual cycle. Do you guys, can I say, this used to be in grade 9 science, and it was a really good, important topic that we used to cover, like human sexual development. And it's not in grade 9 science anymore, but do they at least teach the science of that in like grade 9 or grade 10 health? No? Like, that's ridiculous to me. That actually makes me angry. But like, and I like sex ed class should include a lot of things. Like, it should include talking about like, the politics of sex and the actual like, like the social elements of sex, which are important, right? Like, obviously, very, very, very important. But I think understanding like your biology is also really important. So I think it's ridiculous that the only kids who learn that are kids who take senior biology, and even then, like, we don't actually talk about like the developmental biology of it, right? But like, yeah. So women are born with every egg they're ever going to have in their body. And over the course of their life, from the onset of puberty, with each month, an egg is released into their reproductive organs and is fostered there. And if it's fertilized, then that will turn into an embryo. If it's not, it's released. And that's what your menstrual flow is, right? That's the release of both that embryo and the endometrial lining, the lining that's been produced there to nurture it in case it is fertilized. How long do women keep releasing eggs for? Typically, I mean, there's a massive span, but typically speaking. Yeah, and for most women, it'll be like in the mid-40s that they stop ovulating, though for some it's as early as their late 30s, and for some it's as late as their 50s. There's a big spread of what is normal there, and that's typical in a lot of biological things. What do we call that point when a woman stops menstruating? Menopause, that's right. Which isn't actually just the word menstruating and the word pause, like pause on a TV, like put together, but it kind of works like that's, but it's really more like mena stop because it doesn't like start again later. But yeah, so like women have a fertility window before which and after which they aren't reproductively viable, whereas for men they have a fertility window below which they're not reproductively viable, but typically men will stay reproductively viable for the duration of their life. Okay, look here. Kind of zoomed out, you can see what meiosis looks like on the whole. And the main thing to take away is it's just the same cycle happening twice. Does that make sense? And the only difference happens in anaphase. In meiosis, the first time through anaphase, the chromosomes don't split, they get pulled to each side, still attached to each other. And then in the second time through, during anaphase, the chromosomes are split. And that's really it. So it looks big and complicated, it looks big and scary, but really, you're just drawing exactly mitosis again, twice, and the only thing that changes is in anaphase. Anaphase 1, not even anaphase 2. Anaphase 2 is just like mitosis would be. But this is extremely important because when we get together on Monday and talk about how your genes get passed on, and we talk about the fact that, well, mom will either provide this allele or that allele, and dad will either provide this allele or that allele, you'll understand what it means, which is that every single time a sperm gets produced, it's either getting produced with this chromosome or with this chromosome. And then it's also either, and it's also getting produced either with that chromosome or with that one, but not both. Does that make sense? So your sperm cell could have these two together, or it could have these two together, or it could have those two together and those two together. Does that make sense? Any combination thereof. So there's, from a single cell, there's 4 possible sperm cells or 4 possible egg cells that can be produced. Does that make sense? You can either have mom's chromosome, dad's chromosome, dad's chromosome. But that's the thing. I drew this as having how many different chromosomes? Just 2, right? So for this example, 2 chromosomes. There are 4 possible sperm cells produced. And we could say it's 1 from mom, 2 from mom, 1 from dad, I should say paternal, 2 from dad, 1 from mom, 2 from dad, or 1 from dad, 2 from mom. Right? But again, we don't have 2 chromosomes. Humans have 23 chromosomes. Here's your challenge, and maybe talk to somebody in data management. How many possible sex cells how many possible sex cell combinations can be made from your 23 chromosomes? Anyone here in data management? It would not.
More than one.
It is a lot. A lot is a good answer. Think about it. It's a good data management problem. And think, how did we get this? Not a factorial. No, figure it out. I'm not giving hints. Yeah. And you could just go ask Calford, but that feels like a cop-out. I think you can figure it out. You're smart. How many different ways can you arrange? Because remember, that means we have 23. That means we have chromosome 1 from mom, chromosome 1 from dad, chromosome 2 from mom, chromosome 2 from dad, 3 from mom, 3 from dad, all the way down to 22 from mom, 22 from dad, 23 from mom, 23 from dad. So how many ways can you make one that this is going to get turned into 1 question mark, 2 question mark, 3 question mark, ... 22 question mark, 23 question mark. How many possible combinations can you do that? How many ways can you do that? One way would be every single one from mom. One way would be every single one from dad. Another one would be alternating. Mom, dad, mom, dad, mom, dad, mom, dad. I guess you could do the other way. Dad, mom, dad, mom, dad, mom. How many different ways can you put those together? You can figure it out. You're smart. And don't just Google it either. That's lazy and lame. Work it out. That's all for me today.